Hello everyone. Welcome to another Lunch and Learn by the Florida Native Plant Society. I'm Valerie Anderson, Director of Communications and Programming. Today we have a really great presentation with Lilly Anderson-Messec and Ashlynn Smith, and they've been working really hard to protect and monitor this endangered tree in the Panhandle. Before we get started, I want to say thank you to everybody who's done Sable Minor submissions. That should be coming out in the beginning of July. It's going to be a pretty substantial issue, so keep an eye on your inboxes. We did get Alan Frank, got him committed to the Lunch and Learn next week, so look forward to a presentation entitled 123 Species of Chiacoca and Rubiaceae in Florida. So that I hadn't heard of, South Florida plant. West Indian milkberry is the most common one on iNaturalist, and so, I'm looking forward to learning more. And then the week after, we begin our series on endangered plants with Dr. Patty Anderson, botanist at FDAX. And so that's everything. The first part is everything you want to know about protecting Florida's endangered plants. And the next week is some like hot spots of endangered plant species in Florida. So join us on upcoming Lunch and Learns for some really exciting topics. And without further ado, here is Lilly and Ashlynn.
Hello, welcome to our presentation, Collaboration for Conservation, the Florida Native Plant Society's TorreyaKeepers program in collaboration with the Atlanta Botanical Garden. And I am Lilly Anderson-Messec. I'm the Director of North Florida Programs for the Florida Native Plant Society and the TorreyaKeepers Project Manager.
And I'm Ashlynn Smith. I am the Gulf Coast Coordinator for the Conservation and Research Department at Atlanta Botanical Garden.
And this project is just a really great example of how much you can get done when organizations and individuals collaborate effectively. We have been able to do so much more than we could have done as single entities or individuals. And we've built a whole lot of great relationships and friendships that have come out of it as well. Like Ashlynn is going to be one of my best friends for life from now on, from here on out. So first off, I wanted to just state a land acknowledgement to remind us that the natural range of Torreya taxifolia is found on ancestral and unceded lands of the Muskogee Creek and Appalachee people. They, along with their ancestors, have been stewards of this region for generations. And we also wanted to mention that many organizations and individuals have contributed immensely to the conservation of Torreya taxifolia. There are far too many for us to mention in the time given in this presentation. And this presentation is just, we're just representing the current works of the TorreyaKeepers Project, which is built upon and in collaboration with a lot of contributions from many organizations and individuals. And we wanted to give credit and thanks to everyone who has contributed to the accumulated knowledge and protection of this species. Okay, so introducing Torreya taxifolia, the Florida Torreya. Here's a photo on the right here of me standing next to one of the most large, healthy, and mature representatives found within its natural range. And this is actually, though, a planted specimen that is planted at a state park. So first off, I'm going to go over a few basics. Facts and identifying features of Torreya taxifolia, and then I'm going to mention a few commonly confused species that people mistake as Torreya, which are not. So to start with, Torreya is a genus of conifers made up of 6 or 7 species within the family Taxaceae. 4 species are native to Eastern Asia. The other 2 are native to North America. And Torreya californica is a species that is native to the west coast of the US. The genus is named after the American botanist, John Torrey, and he first identified it as a new species after Hardy Brian Kroom sent him specimens from this area in Florida. And these are small to medium-sized evergreen trees. And although they once did reach 30 to 60 feet tall at maturity, And I just wanted to point out on the photo on the right, the leaf arrangement, which is very indicative to identification, how the leaves are in pairs and flat on the, arranged flat on the branches. The leaves are very needle-like, hard in texture, and very sharp and painful to the touch. So here's another form of arrangement that is found on the tree. On the stems and on the trunks, the leaves are arranged spirally, while on the branches, they're arranged flat. So that's an identifying feature that's good to remember. And they're also, again, very linear, lanceolate, hard and stiff in texture. And then that end, that pointed end of the needle-like leaf is very sharp and hurts. That's why I'm wearing gloves in the picture in the center there. So Torreya species can either be monoecious or dioecious, but Torreya taxifolia, our Florida Torreya, is dioecious. And that means that each individual tree is either male or female. And the male pollen producing cones, which you can see on the right here, are grouped in lines along the underside of the branch. And the female seed producing cones, which they're both found on different separate individual trees. The females are single or grouped 2 to 8 together they are on short stems, and they begin small and then mature. It takes them about 18 months to mature into a drupe-like structure with a single large nut-like seed that is surrounded by a fleshy covering. And then here on the left, you can see in the top upper left corner, that's a male tree maturing its pollen cones. It's just beginning to mature them. And then in the bottom left, you can see a very mature female cone that is close to maturity. And then on the right here, there's a small Torreya, and notice the leaf arrangements on those branches, very flat. And so this is another very rare species that is commonly misidentified as Torreya. It is also rare, endemic, and endangered, and it also occurs in the same very unique steephead ravines along the Apalachicola River, and that is Florida yew, Taxus floridana. The species is possibly even more rare in numbers than Torreya, but since it has always been rare and it hasn't had this catastrophic decline that Torreya has experienced, it's not garnered quite as much attention because of that fact. And so from a distance or in photos, it can be very difficult to differentiate Torreya and Taxus. And I certainly have been fooled out in the field when I see a conifer with that needle leaf arrangement at a distance, and then I get closer and I realize that it's Taxus and not Torreya. But notice here that the needles are slightly shorter than Torreya needles are. They have a more blunt tip. They're not pointy or pokey or painful. And they also, the new growth on the branches have this glaucous coating that give it a matte-like blue-gray appearance. And you can definitely easily differentiate them if you touch them, if you're with the plant in person, because the Taxus needles are very soft and not painful to touch. Again, they're soft and flexible, whereas, the Torreya is stiff and hard and pointy. And like Torreya, Taxus floridana is dioecious with male and female cones on separate plants. And in the lower left-hand corner, you can see a ripening female cones on this Taxus. They look very different than the Torreya cones. So again, the needles of Taxus are arranged very similarly to Torreya. They also lie flat on the branches and spiral on the stems, but they tend to be less orderly in appearance, even from a distance, because the Torreya needles are so stiff that they always look much more like orderly, I guess I would say. The bark is also different. You can see in the photo on the right and the bottom left-hand side of it, is on the left, you see the Taxus bark, which is thin, scaly, and purple brown in color, as compared to on the right of it, the Torreya bark, which is much more paler and furrowed. And so here's a slide comparing the reproductive structures of Torreya and on the left and Taxus on the right. As I mentioned, Taxus is dioecious with separate male and female plants. The female seed cone is highly modified, berry-like, with a single scale developing into a soft, juicy, bright red aril containing a single dark brown seed. And you saw that bright red aril in the last slide. And the fruits occur singly or on a few leaf axils. And the pollen cones are globose and produced on the undersides of the shoots in early spring. And another species that is commonly confused with Torreya, but is actually a non-native species, is Chinese fir cunninghamia lanceolata. This species is in the cypress family. And when someone calls and tells me they have a mature Torreya in their yard or neighborhood, it is always this species. They're very similar in appearance, but these are all planted specimens that would not be found in steephead ravines where you would find Taxus and Torreya. And thankfully they don't reseed, so I'm glad that that's the case since they're not native. They're native to South Central and Southeast China. They were brought here for the horticultural industry, and they actually could be a possible introduction for the fungus that is killing Torreya, which we will discuss in a moment. But a little bit more about the Chinese fir. They're monoecious, so as compared to Torreya and Taxus, they have both male and female reproductive structures on the same plant. And it is very, the reproductive structures are very different than Torreya. The female cones look like pine cones. You can see them on the right here. And then further, just to the right of the female cones, you can see the male cones on the same plant, the male pollen producing cones. And so the needles are very similar to Torreya in that they're evergreen, they're sharp and stiff, but they are not arranged flat on the branches. They're spirally arranged on the branches. So that is a really easy way to identify this species, even when it's not fruiting, because the branches have that fluffier look. And so here's a comparison of the three species we just went over. Torreya on the left with its flat, shiny, stiff, sharp needles, and Chinese fir in the middle with the more fluffy, spirally arranged needles that are sharp and stiff. And then Florida yew on the right with its soft, more blunt edged needles and the same flat leaf arrangement. So getting back to Torreya. Torreya is endemic to the really unique habitats called steephead ravines along the Apalachicola River in the Florida Panhandle. And there are a few populations that occur just over the state line in Southwest Georgia. And the historic range of Torreya taxifolia has always been very narrow. Here are some historical maps over the years. But it was once much more common than it is now and was a very common component of the canopy in these wooded steephead ravines. And there used to be plentiful specimens 30 to 60 feet tall that were frequently harvested for its durable wood by locals. So since the 1950s, the species has been in catastrophic decline. Though these trees were once very tall, like I just mentioned, up to 60 feet, it's now very uncommon to find a tree that's over 10 feet. There's a photo of me on the right standing with a tree that's at least 15, maybe 20 feet tall, probably 15. But that is a very uncommon sight. You typically see them, the photo in the middle here, Nicole, with this small little Torreya. That's much more common to see these small saplings. And because there are so few trees that reach maturity, there's virtually no reproduction in the wild. So here's a timeline of the decline of the species. In 1938, they first observed some needle loss, some leaf loss. And then by 1958, people were noticing dead trees up higher on the ravine slopes. And then by 1962, virtually all canopy-sized trees were dead and only resprouts remained. 1984, it was listed as endangered under the Endangered Species Act, and 86, U.S. Fish and Wildlife Recovery Plan was written for Torreya. In 89, Atlanta Botanical Garden began their conservation efforts on the species. And in 2011, which was a big advancement, was the fungal pathogen that is actually killing the species was named and identified by Dr. Jason Smith at the University of Florida. And in March of 2018, the TorreyaKeepers Project was formed as an outgrowth of the Florida Tree of Life Conference. And in October 2018, Hurricane Michael hit. That was a difficult time. And that hurricane was a catastrophic, Category 5 hurricane, which drastically altered the slope forest that are the sensitive habitat of Torreya, and it caused approximately 20% mortality. So as of 2019, there were just over 700 trees known remaining, and the Atlanta Botanical Garden and Florida Native Plant Society continued surveying. But there's currently still no known recruitment in the wild. So over the years, there have been many theories and hypotheses about why Torreya was declining so rapidly. And it's been hotly debated, but we are certain that habitat degradation due to development, silviculture, climate change, and other human causes are definitely contributors to the decline. The trees also are known to be susceptible to damage from deer rubbing, although that would be something that we would assume they would have evolved to withstand. And what we have realized by 2011 was that the primary culprit for the death and decline of the species is due to a fungus. And it was named, it was a new to science fungus called Fusarium torreyae. So next slide, or no, not next slide, sorry. This is Fusarium torreyae is a fungal pathogen recently identified and described by Dr. Jason Smith at the University of Florida, as I mentioned before. And his lab is continuing to research this newly described species. Jason suspects that this fungus evolved in Asia, along with relatives of Torreya taxifolia native to that region. And it was probably introduced through the import of those non-native species for horticultural uses. And of course, more work will be needed to know for sure whether that is truly the case, but that's what the current thought is. And so when the trees are stressed, the fungus causes leaf blight and stem cankers and can ultimately kill it back down to the root system or kill the plant entirely. And the tree might resprout from those roots if the roots live, but it will remain infected with the fungus. And trees can be infected for many years without showing any signs of the disease. And then they might begin to show signs if they become stressed. And so every Torreya taxifolia tree that has been tested for this fungus has been proven to be infected. And so even if your plant doesn't show signs of disease, it's probable that all plant material of the species is infected with this fungus. And so most importantly and most worryingly, and one thing I want to just really reiterate as one of the most important things we have learned is that for this reason, or for the reason that I'm about to state, that this is likely an airborne species. The spores most likely spread through the air, and it has been proven to jump to and infect other unrelated hardwood species. And also, most importantly, other very rare conifers on the East Coast, such as Carolina hemlocks, eastern hemlocks, and other pinus species. So it's very important that you don't plant Torreya taxifolia outside of its native range, since all known material is infected, and this fungus can spread to other hardwoods and other rare conifers, and it could cause, really terrible unforeseen consequences if it continues to spread. So not only is Torreya facing this, fungus, it also has had to deal with the aftermath of Hurricane Michael. So Hurricane Michael brought devastation to the entire range of Torreya, and it killed many trees and drastically changed the habitat. And the future of the species is in really great peril, and more research on the fusarium must continue in order to find a treatment for the blight. But I just wanted to show, here are some photos of just a tiny piece of the devastation of Hurricane Michael. You can see Ashlynn on the left standing with most of the mature trees in some areas are just all toppled over. And then on the right, there's Ashlynn standing underneath 3 felled trees that have fallen over this Florida yew, which is still kind of reaching and trying to grow up in between them. And then in the center here, I'm standing and sweating profusely on this ravine slope that has multiple, so when all these trees fall, they fall into the ravines. And so these steep ravine slopes are, they're matchsticks of large felled trees. And then the secondary growth from the disturbed soil has been what is the most difficult to deal with, because we have just complete tangles of grapevines and blackberry and all kinds of little shrubby stuff that is coming up and becomes kind of an impenetrable mass. So the TorreyaKeepers Project was originally started by members of the Florida Native Plant Society's Magnolia chapter and spearheaded by Lee Brooks, Bill Booth, and Helen Roth, who are all local landowners in the Torreya range and native plant enthusiasts and experts. They began surveying privately owned lands in this area for undocumented trees and collaborated with the Atlanta Botanical Garden, the University of Florida, Torreya State Park, and the U.S. Fish and Wildlife. And the project was eventually adopted in 2019 by the state organization of the Florida Native Plant Society. And I joined the project working for the Florida Native Plant Society in 2019. So the TorreyaKeepers Project currently is working very closely in collaboration with the Atlanta Botanical Garden to continue surveying public and private lands, especially for remaining trees. And we collect data on the trees that we find. We also collect tissue samples for genetic testing and cuttings, which are then grown by the Atlanta Botanical Garden to preserve the genetic diversity of the tree, which is very important. And we also collect canker samples for research at the University of Florida.
So as Lilly mentioned, there are currently a very large number of conservation actions taking place on behalf of Torreya taxifolia. These actions are conducted by many people and many organizations. So I will talk briefly about each of them, but the majority of what I'm going to be talking about is going to be the private property surveys. But it's important to know that each of these actions is vital to the continued survival of this species in the wild, and it provides information as to how to better, how to approach the protection of this species. And it gives us better understanding of all of the aspects involved. So as I mentioned, I'll be talking about a few of the conservation actions, but primarily focusing on the private property surveys. So Lilly and I lead this effort to survey private properties for Torreya taxifolia. But I wanted to acknowledge that this portion of the conservation work would most definitely not be possible or wouldn't exist without our fearless leaders, Juliet Rainier and Emily Coffey. These women have worked tirelessly for this species. They work to protect rare plants all over the Southeast and in Florida. And Lilly and I would not have the opportunity to work with Torreya without them. So we're just really thankful that they've allowed us to be the local lead for this project. And we really appreciate their constant, unwavering enthusiasm for this species, as well as their support and encouragement that they give to us, on a daily basis. So now I'm going to get into a little bit more detail about the private property surveys. So the first question that some people might ask about this project, certainly no one listening to this presentation right now, but some people might ask, why would you even bother surveying private properties? Don't we have enough of them on public lands? Aren't they better protected on public lands? And, isn't it a big hassle? And a lot of time and resources are required to contact private property owners and obtain permission, line up permits and such. But I would say that there are many reasons why we need to survey for all rare plants on private property and connect with these property owners to help protect those rare plants. So one of the reasons being that specifically for Torreya, this objective was outlined explicitly in the 1986 recovery plan for the species. So here's a screenshot of that document, and it states that private lands should be searched for Torreya trees and cooperation sought from owners to protect trees and allow access for conservation purposes. As this information becomes available from the search for trees, we then need to determine what land is needed to support reintroduction of populations and develop a plan for habitat protection, including cooperative agreements, easements or land acquisition, as well as habitat management measures. Another reason that this is important is because private property owners in the Apalachicola area, they didn't see any less damage from Hurricane Michael than public lands, of course, but they do have, especially for conservation-minded private property owners, they have fewer resources available to repair and restore their properties to, pre-Michael conditions. They also may be conservation-minded and care about rare plants, but not know that they have an extremely rare plant on their property, or other private property owners have different land management objectives altogether. So regardless of the private property owner and their objectives, it's essential for the continued existence of the species that we reach out to everyone who is potentially a property owner with this tree on their property to let them know about the tree, see if they're interested in allowing us to have access to search for it, so that then we can protect it in our XC2 collections, and also just get documentation that the tree is there and basic health measurements. These are both pictures also from private property surveys that we've conducted just to show the extent of the damage. And then sort of a subsequent damage, you can see on the picture on the right is, changes in land management and hurricane damage lead to very severe erosion in some cases on these properties. So property owners dealing with issues like this has just got to be very overwhelming. So another reason why this is important is because, so as we suspected, prior to this collaboration and these targeted surveys on private properties, out of all of the known trees, there were 82% of them occurred on public lands versus just 18% on private. And just one season, of primarily Lilly getting out there and, having boots on the ground, contacting property owners, reaching out to them, we have new numbers of trees. And so now the percentages have shifted a little bit, and there are 66% on public lands and 34% on private. So I would think that with continued surveys, continued reaching out to private property owners, that these numbers are going to even out even further. So just to give you a little bit more information about what's been done thus far for this project, over 600 acres of property, private property, have been surveyed across 6 private property owners, and over 150 new trees have been found. I will say that the private property owners in some cases did know about these trees, but they had not been tagged and data had not been collected on them. So now we've got them in our records. We also have many of them in our XC2 collections. So we collect DNA samples, as Lilly mentioned, on every single tree that's found, because it's just a very small sample. And then on a portion of the trees, we also collect cuttings. So from the 2020 to 2021 surveys, we've added 73 trees to the XC2 collections from private property and 9 from public property. So it just emphasizes the importance of actually reaching out to property owners, making contact with them, letting them know they have the potential to have this tree, and just asking if they mind if we take a look. And we'll talk about some of those collaborative partners later on, but that's what we've done thus far on this portion of the project. It's great work. It's been a lot of fun working with Lilly. I have to say the same. I think yeah, we're going to be friends for a long time. It's just been a great experience with FNPS and with Lilly. So now I'm going to talk a little bit about, I did just want to show also the, just an example. Lilly already talked about this. We know that working in Florida, working, in hot, humid conditions with insects is already tough, but adding this aspect of down debris and the vines growing on top of it, just, it can be tough. So this is a picture that I took standing on top of a log. There's a stream, small stream down here. And then in the picture on the left, there is a Torreya in that picture, but you can't see it until you go to this middle picture. You get really close to it, and then you can start to see the needles coming through. And this is a tree that we had a GPS location for. We were just trying to get to it to assess it and collect samples. So the picture on the right is one of our summer interns, Marcella Johnson, getting the height measurements for that tree after we've removed and cleared the debris off of it. So just wanted to, reiterate that this is not easy work. And so anyone working on this, all the volunteers just deserve so much gratitude from us and kudos, because not easy. Okay. So now I'm going to talk about the other conservation efforts going on. I'm going to just very briefly touch on these. There are people, I'll give you their contact information to the specific people working on these projects, because they are would be much better at answering questions about individual components. So we definitely, okay, so conservation genetics is not my area of expertise, but it's another component and should be a component to any rare plant conservation effort. This is, like I said, not my specific area of expertise, but anyone listening knows already that genetic diversity is strongly associated with the likelihood of species recovery. The ability of a species to be able to resist disturbances such as hurricanes and the resiliency to recover is strongly tied to genetic diversity. So conservation genetics utilizes genetic theory and techniques to reduce the risk of extinction in threatened species. And for Torreya specifically, conservation genetics can be applied to inform us about how genetically robust the populations are in the wild, the remaining trees, as well as how successful we've been in capturing genetic diversity in our XC2 collections. This work is being carried out through a partnership between ABG and the University of Georgia. Below is a list of the results that they've obtained thus far, but that last item on the list is really important. They're developing a probe set to perform sequence capture on 325 Torreya individuals. So individuals that were found this summer on private property are now going to be included in this analysis, which is amazing. If you have any questions about the specifics about the conservation genomics portion, you can contact Dr. Lauren Easterman, and her contact information is right there on the left. XC2 collections are also important because they provide a backup of genetic material in case of local extirpation or extinction. So there are currently over 500 trees represented at ABG in XC2 collections. And so these are individuals, and that number is not representative of any clones. Torreya taxifolia, because of this, it's represented more than any other rare plant species in the Southeast in XC2 collections by far. So these trees are not only providing just a backup of genetic material, but they also have provided seed for other studies. And one of them I'll talk about in a minute. But this area on the slide that's outlined in red is our conservation nursery in Gainesville, Georgia. And it's this area in red is specifically where the Torreya are held right now. But as the number of cuttings increases, the need to store and care for these plants as they mature also increases. And we've received funding from the U.S. Fish and Wildlife Service Recovery Challenge Grant to expand the nursery space available for growing trees at the Gainesville nursery. So, and that's also, of course, a collaboration between ABG and the Florida Native Plant Society, because the cuttings that are collected on private properties and public are going up to Gainesville. So this effort is led by John Evans, our horticulture manager, Brittany Carson, our Gainesville nursery coordinator, and Rashad Tillery, the conservation horticulturalist for ABG. Another component to the success and recovery of Torreya is gathering information on how it responds to disturbance and whether there could possibly be more devastating long-term effects from Hurricane Michael. As we've stated already, Hurricane Michael was a Category 5 hurricane when it hit the Gulf Coast, and preliminary post-hurricane assessments revealed that there was 20% mortality directly from hurricane damage. So because of this, 40 trees were affixed with humidity and temperature monitors, and now there are annual growth measurements collected, as well as other ecological and habitat assessments. And these will be carried out over the next 10 years. So hopefully getting a very long-term data set on how these trees are responding to the hurricane. Leading this effort is Emily Coffey and our newly appointed conservation coordinator, Loy Xingwen. This is a very large-scale monitoring effort. 40 trees is a lot. The terrain to get to the trees is intense, so it wouldn't be possible at all without many, many volunteers. We have monitoring field days again this fall, and so please reach out to Emily or Loy if you have any questions about the specifics of the monitoring, or if you want to help out this fall. Seed trials were also implemented to determine whether planting seed directly is a feasible option for restoration of this species into its native range. So to do this, we selected 9 seed trial locations on both public and private land. We planted 3 seeds from 7 maternal lines in both caged and uncaged areas at each location. The cages were affixed with temperature and humidity sensors to capture the site-specific data on those parameters. And also just to note, the seed was sourced from nursery plants, so this seed was not taken from wild populations. And they were also cold stratified for 2 years prior to planting. And data is still being collected on the germination and survival of seedlings. There's just a picture here at the bottom showing one of those seed trials that was just implemented. And then we have a picture of one of our seedlings emerging from the ground. It germinated and grew. I thought that was a great picture. But as I said, the data is still being collected on that study. Habitat suitability models are also becoming more frequently used for rare plant conservation as a whole. I'm not going to dive into the specifics of these models, but I wanted to provide a bit of information that to people who may not be familiar with them at all. So the premise is that the model is used to utilize the best available occurrence information, pair that with several environmental variables, including soils, elevation, and climate data. There's other variables that you can use as well in order to produce a map of potential suitable habitat. So the process to go from the inputs to the model outputs can vary greatly, as you know, there are many types of models to run. There's lots of software programs that you can use, but the result is the same. It's a map that can be used for prioritization of rare plant searches, as well as determining the most appropriate locations for reintroduction within a species' natural range. So Nicole Zampieri is a regular volunteer doing Torreya surveys with us. She's also a graduate student at Florida State University. She did some preliminary modeling for Torreya as part of a class project, actually, and has provided her data and files to our intern, Nabil Murshid, who will be refining the model and comparing it with other model types to get the best results. So we're all going to be working on this summer. We're going to be refining Nicole's model, but we're just super thankful to her for sharing this information with us and getting things started. Going to allow us to get a lot further this summer, for sure. So in order to discuss the next action, I do need to provide a little bit of background information and give credit to the folks who have been working with the fusarium species infecting Florida Torreya. So Lilly's already mentioned this, but Jason Smith and colleagues identified Fusarium torreyae as a fungal pathogen causing the decline of the Torreya. They were able to isolate it directly from cankers collected from the trees, and they found that 5-year-old seedlings died within 6 to 8 weeks of inoculation. And I'm not going to get into any more of the details about that, but this work is extremely important. They're continuing to work with this Fusarium species, and for questions about it, contact Jason Smith. I've included his email address there below. So because of this great work that they've already done, and they've already been able to identify the pathogen and isolate it, we can now test whether fungicide applications may help Torreya fight the effects of fungal inoculation. So to do this, ABG is using a paper written on fungicide application to treat the laurel wilt disease and red bay. We're using it as a guide to conduct a similar study with Torreya and the fusarium. So this project is just now getting ramped up. It's starting probably in a few weeks, and to get more information about that and leading the study will be Emily, Lauren, and John, and we'll also have assistance from an intern, Ian Kelly. So that summarizes the specific targeted private property surveys, as well as some of the other conservation actions that have been going on with Torreya taxifolia. But we definitely wanted to say thank you, give a special thank you, of course, to the private landowners who have worked with us on this project. And I'll let Lilly take it away.
Yeah, I wanted to say specific things also to the private landowners, because without them, this project wouldn't be where we wouldn't be able to do it. So, and I have been really inspired by how excited and interested and gracious they have been, how interested they are in the species on their property, and just how kind and gracious they've been allowing us to come onto their lands for sometimes weeks or months at a time to survey. Also, our volunteers have been incredible, and Next slide. The volunteer work is not easy. I just wanted to play these little videos, or here's a few photos of some of our volunteers who have been, thrown into the, the viper pit here, basically. We try to warn them ahead of time of how difficult this work is, but it's very difficult work. Not only are the ravines like extremely steep already and difficult terrain initially. Next slide, if you'll show like, I think there's a video of Nicole and Ashlynn here scaling one of these ravines. You're often like laying on your body, climbing on your stomach up a ravine, and sometimes they're muddy and slippery. And then what's even worse is when the matchsticks of felled trees get covered in vines and other, blackberries and all sorts of stuff. And then there's this center video here of Marcella and I in the middle of a whole wide seed of Vitus vines. And we knew that there was a Torreya in there because we had the location, the lat long for it already. And so we just had to go through, some of these areas you're trying to get, there's no road, or trail. So you're just like hiking from the actual highway on, to try to find this tree, and there's no way but through. And it is difficult work. It's also very hot, as you can see here. One volunteer brought a fan, which was a smart move because it gets extremely humid and hot, and it's difficult work. And I'm just so grateful that there are so many people who are interested. It's difficult, but it's also very fun. And you get to see these incredibly unique habitats that you wouldn't get to see or experience anywhere else because they're so unique. They only occur here. So it's also a real treat to see all of the other cool rare plants that occur in these habitats as well. And then finally, we wanted to thank our funders because without the funding, we wouldn't be able to do any of this. So the majority of this work is funded by grants from the U.S. Fish and Wildlife Service and a sub-recipient grant from the U.S. Fish through the Florida Department of Agriculture. And then we've had gaps in funding at times, and we have been so lucky to have donations from members of the Florida Native Plant Society who have gotten us through so we can continue to work. And then also other anonymous donors who are just conservation minded and interested in the conservation of the species. And also donations are always welcome. But I really just wanted to reiterate that this project is just such a success because of the collaboration of so many individuals, agencies, and the history of all the individual and organizational work that has already been done on this tree. We're building upon that. We didn't start these programs, that we're just continuing to further them and take all of the work that others have done and continue to advance. So yeah, I guess we will. You want to say anything, Ashlynn?
I just want to say thanks to FNPS and thank you, Lilly, and thank you for letting me come in,
I would also say it's been such a treat to work with the Atlanta Botanical Garden. They are such an amazing crew in their research and conservation division. And it's almost all female too, I would say. Like our staff is female, their staff is mostly female. And it's just been so inspiring to work with these incredibly bright, strong, intelligent female scientists. And I've just been loving it, so. All right, well, I guess we'll
questions.
All right, I'm popping myself back in here. Thank you guys, that was fantastic. We do have our first question in the chat. Warren Parr says, I live in the Tallahassee area. If I'm interested in volunteering, who's the best person to email?
Yeah, you can email me, and we can put that in the chat. Val, if you could put it in, it's TorreyaKeepers at fnps.org. And so the work is very difficult, and we've we've ended surveys for this season because it begins to become too dangerously hot, and then we are getting these summer rains and it gets wet and slippery, and that can become dangerous. And then also the visibility is extremely limited in the summer, and it gets worse as the summer progresses and we get more and more vegetative, stuff growing everywhere. And we can't take successful cuttings in the summer either. So we'll begin surveying in the fall, like September probably.
Okay, I put that in the chat. While we're waiting for people to ask more questions, I wanted to just thank everyone who is a member of the Florida Native Plant Society. Your membership enables us to administer these programs and pay some of Lilly's salary. We have grants as well. But yeah, your membership is it's very valuable to us. If you're not a member, that's one way you can support the TorreyaKeepers program and our other work on endangered plants. And I'm going to bring over the website. If you'd like to donate, just go to fnps.org, donate, and then you can how many?
Oh, I don't know. I think it was based off of number of chapters.
Oh.
Maybe.
Yeah, we were the third.
Okay.
So we had there's the Native Plant Trust in New England, and then the California Native Plant Society. I mean, they have I don't know how many staff, but 10 or above, much larger budget than we do. So I would be very shocked if they had more, if we had more members than they did.
Yeah.
But we may have, I think we're pretty close to the Native Plant Trust, so we may have, but they're regional, so they're all of New England.
Yeah. Florida has got just amazing native plant enthusiasts. It's phenomenal.
Oh, we do have a question. Leslie McConaughey asks, what other geographic areas are volunteers concentrated in?
No other geographic areas, because it's a very limited, narrow range. So it's only these steephead ravines that are feed into the Apalachicola River. And so that's Gadsden and Liberty County. There was a population. I mean, there's also, a couple populations above the county state line in Georgia, sorry. And then there was a population on the western side of the river, but that's believed to be extirpated. It's on private land, and it was likely lost due to silviculture, which is tree farming.
So people, if they lived outside of Torreya country, they could volunteer, but they'd have to travel to volunteer.
Correct. They would have to travel down here, and then that's quite
general volunteering. If you wanted to get involved with the horticulture side of Torreya, you could volunteer in Atlanta or in Gainesville, Georgia with ABG. And those, our website has a volunteer form. You can also send an email to me and I can get you hooked up with the folks in Atlanta and in Gainesville.
And again, I want to reiterate that the consensus scientifically is to recommend not to plant Torreya outside of its native range because that this fungal pathogen, which is suspected to be in all Torreya trees existing, can spread to other rare conifers like other hemlocks and pines, and even unrelated hardwood species, and could cause unforeseen cascading effects that could be very negative. So that's just a reminder to plant Torreya only within its range.
Warren Parr asks, are you aware of any conservation programs for the Florida U?
Do you want to answer that, Ashlynn?
So I'm not aware of any large scale ones like this, but something that we've started doing, Lilly and I, in talks with Juliet and Emily, is that when we apply for permits when we're on private property, we include you because we want to go ahead and get a sample of genetic material, and we want to have a permit in hand in case we find one. We can get a cutting and send it up to Atlanta to be propagated. We don't officially have funds to do that yet, but we're on the site. We're taking advantage of being in the area already and having permission from the property owners. So as far as I know, that's on the ABG and FNPS side, that's all that I know of.
But we do, that is a goal to get funding for the Florida U, because it possibly in numbers less plentiful, less common than what's left of the Torreya. So it's always been extremely rare. It just hasn't gotten much attention because it didn't just suddenly decline the way Torreya did, but it is definitely a very rare species that is struggling, and especially struggling after Hurricane Michael. So yeah, so hopefully we'll be able to get funding for Florida U.
Yeah.
And it is listed as state endangered, but not federally endangered.
That's probably another, difficulty in finding funding for it.
Right.
It probably should be, federally endangered, but, it would need to be petitioned.
Well, I mean, how many species have been listed recently federally? Any?
I'm not sure how many. I mean, there's still, is Niger, the area called Niger bractatum, that was recently listed.
It's under review, I believe.
Oh, okay, so that's not even.
For federal listing. Yeah.
It seems strange that the U would not be, I guess it probably just maybe needs more advocacy, because it is very limited both in its range and in its numbers. And that's what, are required to get it listed. And it's very slow at reproducing. Its slope habitats are imperiled by erosion and other issues, and Hurricane Michael. So it should be listed.
Well, lots of kudos in the chat. Bravo.
We just answered everyone's questions then.
Yeah, I think you did.
Yeah.
Well, okay, so we have Lily's email in the chat. Ashlynn, would you like me to put away for people to contact you?
Yeah. It's A.Smith at atlantabg.org.
Okay. Ashlynn's email. All right. Well, this has been, really fun for me, and it looks like people really enjoyed it too, the audience.
Good.
So.
I hope so. It's been a really inspiring project to work on, and it really has been a lot of fun. It's definitely hard work, and there's a lot of, painful times, but it has been overall wonderful experience, and it's given me some hope for, the survival of this species.
Yeah, it's the poster child for what can be done when people, work together on something.
Well, thank you guys for your time and effort and preparing presentation, presenting, and all of your hard work to prevent this species from going extinct. So,
thanks,
Valerie. Thank you. And I hope everyone has an excellent weekend. And if you're not a member of the Florida Native Plant Society, join us.
Yeah.
All right. And with that, we're out.